I thought I did. Woo! Yeah, there I am. Hello.
Hello? Oh, okay. Welcome everyone to Reef Campus again. Just saw some of you earlier this week, multiple times this week. We've had lots of events. So thank you, Mark and Jenny, for coming out to all the events this week. Thank you all for being here tonight as well. We're super excited to have Carlos and Allison Estape here to talk about all of these wonderful fish they've been capturing. It's been a while since Carlos and Allison have been here, but I know all of you are very familiar with them. They always give great presentations and they're avid reef supporters. So we're happy to have them here tonight. It'll just be a normal Carlos and Allison talk with questions at the end. If you guys have questions throughout it, we just ask that you either say them loudly or have the speakers repeat them for the people online. So if you want to refer back to this talk later, you can watch it on our YouTube or Facebook. So it will be recorded for other people to view and for you guys to look back on as well. Before we get started, is there any community announcements, anything like that anybody has? If not, we can just get started. Reminder. Oh, yes.
This coming Wednesday, we're opening a new exhibit, the History of Diving Museum, Aquanauts to Astronauts.
Oh.
A lot of cool stuff. We'll have an astronaut to speak, and we have Kathleen Sullivan's plate suit.
Wow, very
cool. This Wednesday at 5:30.
Okay, this Wednesday at 5:30 at the History Diving Museum.
Correct.
There will be Aquanauts to Astronauts talk. Very cool. And there a new exhibit, and there will be an astronaut there. So why wouldn't you want to go talk to them? Another thing, as a reminder, iCare's Trash Derby is Saturday, tomorrow. It will be in Founders Park. The festival will be. So come out to that. Good time. Lots of trash. Who doesn't love supporting ocean conservation, right? And with that being said, I'll let you guys get started.
Does it pick up my voice at all if I just leave it there? Welcome. This feels like I'm in my living room with all my friends. That's what it feels like to me, coming back all the way back. Tonight's talk is going to be led by Allison. She put an enormous amount of work in this. She had to herd all the scientists who had something to say about all their findings. And so she would have everything ready to go. And then they would say, no, I don't like that word there. And she'd have to move it and stuff. So basically, my job today is going to be color commentary and crowd control. Everybody turn your phones on vibrate or silent if you would. And I think it's time to go.
All right. So welcome. Like Carlos said, we're so excited to be back here. It's Old Home Week, and it's wonderful to see all of your faces in the audience. And as you know, we've been gone for about 2.5 years now, and we've really gone down the whirlpool looking for fish. And this is where we've evolved to. So today we're going to be sharing with you our latest and greatest adventures. And it's a scientific expedition. And we were going to the Revillagigedo Islands. And so this is our logo for the trip. And we're going to explain why this fish is our mascot as we get into the presentation. So the very first question a lot of people have are, where are the Revillagigedo Islands? I've never heard of those before. So for those of you here, we have the East Pacific. You've got the Revillagigedo Islands, more commonly known in the diving community as Socorro, San Benedicto, and Rocca Partida, okay? Because the liveaboards go out there to go see the megafauna. These are the most western islands in the East Pacific. So that's one of the things that was attractive to the scientists for wanting to get out there. Now, it's open ocean, very high energy environment. And this is the Pacific as well. So you've got that huge open ocean. You build up large swells, lots of very high tides, big waves, strong currents, strong winds. In fact, the conditions were pretty challenging at 2 of the islands that we were at. We weren't able to get into where we would like to for all the different habitats because of the surge and the waves. So it's very different diving than here in the Caribbean. That's one of the learning processes that we've had being over on the Pacific side. For all of us here in Florida, we're very familiar with this map. This is the hurricane track map for the last 150 years. The yellow tracks are strong hurricanes. The red are the ones where you're not sure your house is going to be there after it goes by. And over on the left, we can see that the Revillagigedo Islands are in that same track on the Pacific side. So they are getting pummeled throughout the year. And in fact, they have corals growing over in the Pacific, but instead of growing on dead coral platforms like we have here in the Caribbean, they're actually attaching to the hard rocks because of all of that activity with the hurricanes coming through and the environment over there. So why would anybody want to go to the Revillagigedo Islands? Well, it's a UNESCO World Heritage Site, and it's a heritage site because it's one of the best places in the world to see sharks and manta rays. That is their claim to fame. After being there, I can highly recommend it if you want to go see sharks and manta rays. All right, it's a 4-island archipelago. These are all volcanic islands. They're uninhabited except for 2 small military bases. So those guys are really happy when anybody shows up to say hello. All right, and this is Socorro Island. So this was the first stop on our expedition. It's the largest island, about 50 square miles. To get there, it's 300 miles from the tip of Baja. So it took over 38 hours in the liveaboard to get there. So you board the liveaboard like 5 o'clock, they give you dinner, the boat leaves, all the next day you're underway, all that evening you're underway, and it's not until the next morning that you actually arrive in Socorro to be able to go diving. This is the emerged summit of a massive, predominantly submarine shield volcano. And that volcano comes up over 3,400 feet from the bottom. So again, they're open ocean. You've got a lot of water moving around these islands. Now, since we were on a scientific expedition, one of the things that I thought was pretty cool was that this mountain is actually named after an American ichthyologist, Barton Everman, who was promoting research there in the early 1900s. So this area has always been of great interest to the scientific community. We logged 6 dives over 2 days, and we also got in a couple of snorkels. Remember that surge I was telling you? I mean, just watch the fins and the bubbles. So for those of you who take pictures, especially macro, trying to stay still to take a picture of a small fish that's tucked inside, we were getting thrown around. I mean, we'd get back on the boat at the end of the day, and one side would feel longer than the other from the surge. And at the base of those steep walls, you had these huge boulder fields, which was very interesting topography, very different than what we have here. And the fish would just dart in and around those boulder fields. And to sort of give you a little bit of a hint of what's coming, this is on our first island, our friend Alistair finds and photographs this pike blenny. This was the first time this entire genus had ever been documented in these islands. So one of our photographer buddies is already documenting things that the scientists and all these other 150 years worth of coming out to these islands to do research had never found before. So we were very stoked. All right, this is Clarion Island. Now, what makes Clarion so interesting is it is the most western island in the East Pacific. It is extremely remote. Not only is it remote from the mainland, but it is as far from San Benedicto, the very first island, all the way out to Clarion. So it's as far from the mainland. So you've got to go 400 miles to get there from Baja. And from San Benedicto, it's 267 miles. So it is extremely isolated. It's very small, only 8 square miles. It took another 30 hours to get there from Socorro. So those are the kinds of distances that we were traveling. And because of that remoteness, it's rarely visited. So the scuba liveaboards don't go there. In fact, the whole reason this trip was created is Dr. Sanchez, who I'll introduce you in a minute. He's the Mexican scientist. He's like, we really want to get out to Clarion. The only way to do it is to have a private charter. You guys know some photographers. Can we put this charter together so we can get scientists and photographers out there? And that's what led to this whole expedition being created. We were able to log 8 dives and 2 snorkels over 2.5 days. And we were doing a lot of time in the water out there. All right, so again, here we have the large rubble field at the base of the cliffs. You had a lot more sand out there that we were able to get into. Again, diving in as many habitats as we could possibly find. You can see some of the coral growing on the rocks there. And this is the reason we weren't able to get to all the dive sites. So you can see that very steep wall, very dramatic, but look at the surge up top. I mean, no one was getting close to the top because you would just get beaten up. So that was some of the conditions that we were experiencing. And another little hint of what's to come, we found new species, never been documented, never been found before. And so we're going to tell you a little bit more about that later. Okay, this is Roca Partida, very famous within the diving community. It's the tip of a volcano. It's only the football length, American football length in size. And it comes up 120, or goes down 120 feet, very sheer walls. There's a little plateau, and then it drops another mile. So open ocean, very fast currents. We logged 2 dives there. And this is what it looks like. So you can see that very steep wall. And then, I mean, the sharks, I mean, they didn't care. They would just literally come over your shoulder. And what I love is if you watch this school of Creole wrasse, they're coming around and they're just getting thrown around and they can barely swim. I mean, that's what the kind of currents that we were facing. And what's super cool is this next one. So this video, I'm gonna let Carlos share.
Do some color commentary. So we get down, we jump off, and we get down, and one of the very first things we see is like a 5-foot long, am I exaggerating, Jeff? And they were there with us. This maybe bigger. This thing was probably in a sushi market worth about as much as a brand new car, right? I mean, these tuners are very sought after in the East. Anyways, this thing goes by us. I don't get a good photo, and drove me crazy. But this fish wasn't like just leaving. It would just circle and circle and circle Roca Partida. So I timed it. Okay, every 12 minutes or so, the fish would come around. But the second time it came around, it saw me and it went over my head. I said, all right. So we did a second dive. And the next thing that I do is I had one goal in mind, and that was to get a decent photograph of this animal. So I hunted it. I kind of hid in the shadows, waited for it to come out of the sun. And as soon as it did, I got my shot. And that photograph is not mine. This is Ellie, a past reef intern. She got a really good photograph of it. But this is heart-pounding stuff, mantas, silver-tip sharks. I'll let Allison keep continuing.
All right. Yeah. So this was like a merry-go-round, except the fish were on the merry-go-round and you were just standing there watching them go round and round and round. Yeah. Yeah, well, it was. And then this is San Benedicto Island. So this one's about 4 square miles. It's the closest to the mainland, about 250 miles away from Cabo San Lucas. It is a volcano, so you can clearly see the volcano here, all right, and you can see where it erupted. Here's the lava flow down here. This thing erupted in 1952, 1953, wiped out all the flora and fauna on the island. And if you're into interesting tidbits of facts, it exploded at 9 a.m. on August 1st, 1952, and caused the endemic San Benedicto rock wren to go extinct. So the claim to fame of that bird is that we know the exact date and time that the volcano wiped it out. So what that is, you can see since the 50s, you haven't gotten a lot of growth back, but the birds are back. So they really love that island. Here, it's famous for their sharks. So you have the silver-tip sharks, the Galapagos sharks, the white tips. So in the background there's a Galapagos. You don't see the white on the tip like you do with these silver tips. When the video goes through again, you're going to see a flash. That's actually Carlos taking this picture. So you can see the color when you bring your light to the party. And Carlos is going to tell you the next picture.
Well, this is like a flat area, and we got all these sharks on the perimeter. So they tell you, the perimeter is where all the interesting things are, and we're in the middle of this field, and a manta comes by, and then the next thing that comes by is a tiger, a big tiger. I mean, that thing was probably twice the length that I was. Exaggerating? It was a big fish. So, I mean, that was my first tiger shark. It only took me 3,446 dives to finally see one, but there it is.
And I can attest he was swimming towards the tiger shark.
I'm not right in the head. Not right in the head.
I was videotaping it for the insurance purposes. Okay. But yeah, and quite a few of the folks on in the group had some pretty close encounters with these tiger sharks. All right, the other thing they're known for are their very friendly mantas. These guys would literally come up to you, they would look at you, they'd swim away, they'd turn around, they'd come back. We had some just phenomenal experiences with these giant mantas. So if you're into sharks and giant mantas, Can highly recommend this destination. And you get away from the island. I mean, it gets deep fast. You don't have to swim very far. And you'd have schools of tuna. Now, how many of you guys have seen the skipjack tunas that come flying through, or the tunas that come through here? I mean, they move fast, right? So it's like a flock of birds just going so fast you can't even see them. Well, we're on a scientific expedition trying to document the species. So it's not just, oh, look, I got this great picture of a school. We had to go back on the boat, zoom in, and start looking, are there multiple species mixed in with this school? And it actually turns out that there were. We had both frigate mackerel and the black skipjack tuna mixed in. You know, at the time, it's just like a silver blur going by. So at San Benedicto, we did 7 dives, 1 snorkel last November. I mean, the geography is really very interesting underwater. And on the very last dive of the very last day on our 10-day trip, we were able to collect the holotype of another new species. And for those of you who aren't familiar with what a holotype is, it is the single specimen that represents that species for the entire species. So it's a pretty important specimen. And we'll have a great story to share about just how stressful capturing this fish was. All right, so I've told you why scuba divers want to go out there. Why do scientists want to go out there? Well, it's all about geographic isolation. So I already mentioned how the Revillagigedos are 250 miles from the coast of Baja, but it's another 2,600 miles across open ocean before you get to the next island, and that's the Hawaiian Islands. So that's a huge expanse of open ocean. There's no islands, there's nothing out there for the animals to find protection from. And in fact, not only is it wide, it's very deep. So it's over 4 miles deep in some places. So this is really considered a barrier. And because of that, there's very few marine species that can actually make it across this barrier. So if you are a pelagic species like this wahoo, you've got a shot at being able to swim that far. If you are a 1-inch Revillagigedo barnacle blenny, that's a long way to swim to be able to make it across. All right. So because of this, this makes the Revillagigedo Islands absolutely perfect to study speciation and genetic connections. So the scientists are very interested in seeing what's happening with the genetic connection across the East Pacific barrier. Our species crossing is DNA still being transferred. You can also look at it between the islands. I mean, you've got over 250 miles between the island on the far eastern side versus the Clarion on the far western. So is there enough distance there for speciation to be happening? And then you have the same thing happening between the mainland coast of the East Pacific and the islands. So a very, very isolated area that really brings a lot of opportunity to find new species and to see what's happening with speciation. So what kind of speciation are we specifically talking about? It's called allopatric speciation. And this occurs when groups are geographically isolated, and over thousands and thousands of years, hundreds of thousands of years, that isolated population is just reproducing within itself. And because of that, you have genetic divergence happening and you get new species. So what does that look like? So for example, this is the barred serrano. This is its geographic range. So the yellow is the range and the red spots are where it's actually been collected. So you can find the barred serrano throughout the entire East Pacific, including Galapagos, except you won't find it in the Revillagigedo Islands or Clipperton Island or Cocos Island. This is the Socorro serrano. So this is the one on our t-shirt. You can only find the Socorro serrano in the Revillagigedo Islands. You won't find the barred serrano there. So you have genetic divergence, new species developing. And then this is the Cocos serrano. Only place in the world you can find it is at Cocos Island. So this is a great example of how geographic isolation has enabled these different species to emerge. And this is what we were looking for. So prior to our going to the Revillagigedo Islands, there were 13 documented endemic species, which means the only place you find them in the world is right there. So these are examples. You can see most of them are small. They're 3 inches or smaller. The Socorro soapfish can get almost 10 inches in length. So that's not a very big fish. This is our Socorro Serrano, and this is really special to us because this picture was taken by Dr. Ross Robertson, who was one of our scientific trip leaders. And he was there in the 1990s, and he found this fish. That's his picture that he took from 1992. So imagine film. Back then, you're 36 shots, all right, and you're trying to catch a new species that's not sitting still for you. So he got the picture, he collected the fish. It's a new species. He described it. And so for him, we were going back, and we thought that this was a perfect mascot for this trip. And he actually described 3 different species from the Revillagigedo Islands that he was able to collect in his previous expeditions there. So the question for us was, would we find new species, and if so, how many? And that was one of the key motivators for quite a few of us to get out there. All right, now let's talk a little bit about what does it take to describe a new species today. So in the taxonomic gold standard used today to describe a new species, you want to have an in situ diagnostic image. So you can see what the fish looks like in its native habitat. You can see its color patterns. You also want to collect the fish that you took pictures of so that you have those actual specimens, and you can relate this specimen back to what it looked like when it was alive and you had these different colors, and you want to get quite a few specimens. You need to do DNA barcoding, so that way we can look at the genetics and make a real fair comparison on how similar is this fish to some of its other cousins. And then you need to look at the morphological differences, and the morphological differences can be everything from number of scales, fin rays, color patterns, and see how do these compare, how consistent are they, or how divergent are they from the other ones out there. So this is the current gold standard for describing a new species. Now let's take a step back and go back 140 years to 1889. Charles Gilbert was out in the Revillagigedo Islands collecting fish, and they actually collected quite a few specimens. They got 3 specimens of this black wrasse, and at the top, that is their scientific description of the black wrasse. That's my picture. It's the only individual we found in our entire trip out there. And you can see that his description still is a great description of that fish. It hasn't evolved much since 1889 when they captured it. So you can see we've evolved today, now including underwater photos, including the DNA, to the new taxonomic description. All right, so explained why people want to go out there, explained why the scientists want to go out there. What made this expedition different than previous scientific expeditions? Well, on our trip, we combined the skills of marine scientists, marine taxonomists, and underwater photographers. So here on the left, this is Dr. Carlos Sanchez. This is actually him doing the merry-go-round at Roca Partida. And he was our trip leader. He is a Mexican scientist and professor. He was the one that was able to secure the permission from the Mexican authorities for us to go in and do the work that we did and go where we went. And he obtained the permit for us. And on the right is our good buddy, Andre, and he is one of the photographers, and he is a phenomenal photographer, and he can find cryptobenthic species. And so he not only finds them, photographs them, but collects them and brings them back to the scientist. So we were combining the skill sets of both of these different, I guess, sides of science. And so here we have Andre down there taking pictures. And for those of you at Reef, you're going to recognize some of the ladies in these pictures. So on the left, we have Ellie Place. On the right, we have Carrie Kinning, both Reef interns, both past Reef employees, both very good at finding fish. And Ellie actually found this half-banded stargazer. So imagine, most people go there to see sharks and manta rays, and we're finding a 1-inch fish that lives underneath the sand. And this fish actually lives under the sand, so Ellie had to slowly get it up out of the sand so she could get a diagnostic image, shows the bodies, shows the color patterns, took the picture, collected it. So now the scientists can go do the DNA. They can look at the animal and say, hey, do we see any divergence here, or is it the same species that is being found up and down the East Pacific? The other reason you want to have a bunch of photographers on the boat is we're just crazy. All right, so on the left-hand picture, this is not a seal on the far left, that dark figure, that is Andre. And on the right, we have Sarah. And so I remember I told you about the surge. I mean, there was a 6-foot surge coming in. So these guys were timing it, letting the waves take them up, dump them up on the rocks. And they've got their heads stuck in the tide pool because they're looking for clingfish. And that's the clingfish in the center. Now, the scientists are on the boat going, I've never seen anything. You guys are nuts. No scientist would do that. All right. And so every day we did snorkels so that we could get up there on each one of the islands collecting these clingfish and looking for the blennies because, you know, those guys don't swim far. And so there was a high probability that they could potentially be showing speciation. And for those of you who aren't familiar with clingfish, on the far right, you're actually looking at the bottom of the fish. The ventral fins on clingfish have evolved into suckers because they're living in this surge zone. They're getting thrown around all the time, and they're literally able to suck or cling onto the rock and not be blown away like the photographers were. All right, the other reason you have us along are to take high quality in situ diagnostic images. So you've got examples from all of the different photographers. Jeff is here. He and Ann were on the trip with us, lending their fish finding and photography skills. So you're going to see a lot of Jeff's photos featured up here. We were also trying to capture different members of the same species. So if you saw these 3 pictures, you think, oh, these are different species, right? Same species. So look at the variability that you can have in the color patterns. And because the variability is variable and it's not consistent, you know, that's why you can't start breaking them down into different species, because they can have all this different patterning and it's not consistent. We also wanted to capture all of the different development phases. And these 2 images on the left are Jeff. So the beautiful juvenile clarion angelfish. The initial phase in the center, you've got a beautiful orange phase over on the right that our buddy Andre captured. And then Sarah got this gorgeous yellow version. Okay, so again, we wanted to try and capture as many of these different individuals, different color patterns, so that we could create a library. The other thing we wanted to do is we wanted to create and document by island which fish species were found there on that particular day. So all 3 of these fish, first time they had been documented on that particular island. They were known to be in the island chain, but not necessarily on that island. So Jeff got the saber-toothed blenny, Ellie got the barred filefish, and then Lee Richter actually found the big rough scale soldierfish, and we're going to talk about that some more in a minute. All right, so this is the group of photographers and scientists. So if they've got a P by their name or a V, they were a photographer, videographer. The S's were the scientists. You could see all of the stuff that we had to take as part of this expedition. And this was at the very beginning, so we all looked really nice. Yeah, so I didn't look quite this pretty towards the end of the trip. All right.
Go back. Can I say something? Yeah. So imagine this is a liveaboard that's really meant for scuba divers, recreational divers, go out on the islands. This is the upper deck, what I call the Lido deck, and the Lido deck is basically where people go out and sun themselves, hang out, you know, have a few drinks and stuff like that. This was the processing center. This was the lab for all the scientists. This is where all the smelly, bloody, dead fish were at. This is where they got processed. This is where they got labeled. And this is, you know, what you're seeing here. This is ethanol, formaldehyde, not formaldehyde, quinate, all these other different things. So you have to be careful what you grab to drink, whether beer, quinate. So.
Yeah, we actually had to pay a cleaning fee because we brought too many fish scales on board. That's right. All right, so now they've been forewarned. Now they're like, oh, you're coming? All right, this is how much it's going to cost. Okay, so a little bit, to get to know our scientists a little bit better, we had Dr. Ross Robertson from the Smithsonian Tropical Research Institute. He's one of the top marine taxonomists, fish taxonomists in this world, this part of the world. Dr. Carlos Sanchez here on the bottom from the Universidad Autónoma de Baja California Sur. So they were our 2 scientific trip leaders. We had Fernando Duarte, who was getting his master's with Dr. Sanchez, so he got to participate in this. And we also had Dr. Ben Victor. For those of you who don't know, this is Ben Victor of the Ocean Science Foundation. He's doing all the DNA barcoding of life project for reef fishes in the Caribbean and in the East Pacific. So he was very interested and involved and wanting to be part of this so that we could get all those extra missing DNA barcodes. And we had Dr. Michelle Geither from the University of Central Florida. So shout out to one of our Florida universities. We're going to share her work a little bit later. And we also had Ben Frabel and Bill Lutt. Ben was from Scripps Institution of Oceanography, and Bill was from the Natural History Museum in Los Angeles County. And for me, it was really interesting talking to them because they are curators of the fish specimen collections at these two institutions. And I was like, wow, I don't really understand this. And so I got a really great education, and we'll share why their work is so important and how it's contributing to the scientific knowledge base. So wonderful group of scientists. And then here are our photographers. So you can see all of us eager beavers, so super happy to be there. And it was a fun group, all right? A very fun group. All right, so what were the goals of this expedition? It wasn't just go have fun. We wanted to document fish species at each island via photographs, videos, specimen collection, and then the DNA barcoding coming out of that. Second objective, we wanted to create a library of high quality, in situ, taxonomically diagnostic images of all of the reef fishes that we found. I mean, so every island, it was take a picture of every fish you saw. So every single one, we had to document everything we saw. We wanted to obtain specimens for the museum collections and for research. We wanted to document fish fluorescence. We also wanted to gather data on the diversity, abundance, and dispersion of fish eggs and larvae via zooplankton collections to understand, where are the fish coming from? How are they repopulating the islands? What does this look like? And finally, with Dr. Gaither, she was wanting to look at determining the diversity and genetic connectivity between the Revillagigedo fish populations and other fish populations in the East Pacific Islands. So a lot of work, a lot of science was being done. So let's go one by one. So first, documenting fish species via photography. We documented 56% of the reported species in the island. And I say reported because what you do when you go on these trips is you pull up all the previous scientific papers and see what did they report. And some of them say, we saw it, all right? So how scientific is I saw it versus I collected it or I took a photograph of it? So part of the work that's going on right now is trying to validate where do we have vouchers that are either with some sort of physical documentation, either a photo or a specimen to say, yes, this truly was documented on that island. And we're also waiting on all the DNA to come back. So maybe we have some different DNA in there that we haven't seen yet and different species mixed in. So 56% doesn't sound like a lot, but Carlos and I, it took us years and years to get to that number of representation in a different area. So for a 10-day trip, this was a pretty phenomenal result. Now, out of all of the 152 species that we have documented as of today, 7 of those had never been documented before in the Revillagigedo Islands. And out of those 7, 2 to 3 are new species to science. 100 species were documented on different individual islands that had never been documented before. So this is really a lot of information being added to the knowledge base. And we're going to go by island because we have to do this by island. So if you remember, this is Socorro. This is our shield volcano. We were able to document 54% of the island of the reported species. And here is our pike blenny, our claim to fame on that island, being able to add this genus now to the Revillagigedo Islands.
Can I say something? The sad part about this is that pike blennies are so common in pretty much anywhere else if you know how to look for them, that it wasn't collected. We didn't collect it. When Alistair took the photograph and comes back and shows it to the scientists, they had a hissy fit and a conniption together at the same time. So it was like, oh, we have to go find it. But it was the last day that we were there at Socorro. We even talked about returning and blowing off San Benedicto just to go look for this fish, but we never did. So it's going to be another scientific expedition that needs to go find, collect, and then determine what this was.
It's potentially a new species.
Yeah.
All right, so this is Clarion. Again, this one is the most western island in the entire chain. This is the rough scale soldierfish. This was found by Lee Richter. This is a very shy fish. It's nocturnal, so during the daytime, it's hanging out in the caves. Dr. Victor believes this is the first live photo ever taken of this fish in the East Pacific. So that was a really big claim to fame, and we were super excited. Lee found it, comes back, shows everybody. We're all on the second dive. Carlos is like, take me to it. All right, so.
Did I say that?
Yes.
I did. I said that.
The competition amongst all the photographers, we all wanted to get every single fish for our own personal library in addition to contributing it. So it was quite a bit of fun. All right. At Roca Partida, we were able to actually get 60% of the previously reported species. As I mentioned, Ellie was able to document the barred filefish for the first time on that particular site. And again, here you had mainly the pelagics. And then at San Benedicto, the one that we no longer have the endemic rock wren, we were able to get 74%. And that's a pretty significant accomplishment in just 2 days. And here, Ellie again does a fantastic job of finding the Pacific leopard flounder, first time it had ever been documented on that island as well. All right. Now, we also photographed at least 7 species that had not previously been known. I've already mentioned the new genus, and 2 to 3 new endemic species. So right now, I am sorry to say that this is all I can show you of this new species. In the publishing world, this is very competitive. So until the DNA comes back, until this paper is actually published and introduced to the world, we are not allowed to share the actual photos of the fish, because you could see it, you could say, oh, I see what's different, and go publish tomorrow. So this is being held deeply in secret. That is what the fish looks like, so I can at least show you that, the shape. And what I can share with you is this is what Lee Richter looked like when he found it, realized it was really different from all of the others that we had seen, took a picture, collected it, so he's celebrating like crazy underwater.
So this is at Clarion. This is at Clarion. We are, and he shows it to us on the penultimate dive, and he goes, we have to find it, because he shows the photographs to the scientists that go, this is not the other fish in that genus. We need to go collect it. So we didn't want Lee to suffer what poor Alistair had, where he finds the fish and we have no collection, and we really don't know whether it's a new species or not. So what we do is we are on a hunt. Everybody jumps in there, knives in their teeth. We're looking for it. I find it. I take a few photographs of it. I know this is it because I have seen it. I bang my tank and I am mobbed. I am literally pushed out of the way. Okay, so we take a few photographs. The fish is incredibly enough just kept going into this little hole, and then would come back out again and go back into the hole. So it wasn't like swimming away. So it's a fish this big, and we were able to capture it, and it was a big success. So now we're waiting to hear back from the DNA analysis.
All right, so this is what it looked like. So we had bags. You would put your name on it. You would put which number it was so that we could take a picture. Now, so you take a picture of the fish in situ, you'd collect it, you'd take a picture of the bag underwater, so that way we knew that photo went with that fish. And then we'd bring it back, and so the fish got rushed. So we're coming in on the lower boat level of the boat, so you got to rush up steps to get up to the dive deck. Then you had to rush up another flight of steps to get up to the processing center. And we're all running up with these bags in our hands going, look at what we found, look at what we found. Hand it off to Ross and Ben. And then it's like, well, what do you think? Is it different? Is it different? So super exciting for those of us on the photography side in particular who've never been part of this type of experience.
Do you ever worry that you're collecting the last one?
Yeah. Well, I'll show you some photos and we can get into angst in a minute, because we had some of that.
Is there a name for him yet?
Well, that's the scientist that publishes the paper gets to name it. And so we don't know that name yet.
Now the pike, Letty, that's also new, right?
Well, it's definitely a new genus documented for the first time ever in the Revillagigedos. They have to collect it to do the DNA, and then they also have to look at the morphology, because if you don't have both of those together, it's not a new species.
So, but it's new to the island or new?
Well, we don't know that because we don't have a specimen.
So, but it has a name already.
Well, it's being called, well, we don't know which species it is. So we're just calling it Cynopsis sp, because it's never been collected. And so until it's collected, until the DNA is done and the morphological assessment is done to see, does it look different? DNA is different, and it
Mark, it
looks the
remains a
same.
mystery. We don't know.
It's a pike, but it's got a common name. Like the pike.
Well,
No.
pike. Well, the first
Well, it's a
name,
pike, Lenny,
but
we
not. It's
don't
not
know
like
what's-
it's not Mark's pike plenty. We don't know that yet.
It hasn't got it all.
Yes. So opportunity. Believe me, there's scientists out there. They just did another trip recently, and they were going for that fish. All right. So now I want to move on to the holotype of the other species. Now this was interesting. This is a wrasse. One of our previous reef members had actually taken a photo of it way back when, sent it to Dr. Victor. So they knew there was this really weird looking wrasse, but the photo wasn't that great. That was the only image out there. So I was like, we think there's a new species, but we're not sure. And it was a 6-inch fish. That's pretty big for a new species. So we're all out there looking and looking, and it's the last day, and we get out there and we find it. Or Dr. Sanchez sees the initial phase. So he's out there, he's on the first dive, he sees it, comes back to the boat, says, I saw it. And now this dive, this is where we showed you that tiger shark and the other shark video. So we're at San Benedicto. So on the first dive, the photographers are all out there taking pictures of the sharks and the manta rays. And so we come back, Dr. Sanchez says, I found the mystery wrasse. This is what it looks like. So he's giving us a search image on this. At that time, he only saw the initial phase. So it was only 3 inches. He goes, it's not over there by the sharks. It's over there by the rocks. Second dive, all the photographers, our job is to find that fish. And Jeff and Ann can tell us we were swimming like wrasses in between the rocks, up and down, all around. I mean, just you came back dizzy because these things, running into each other, running into the rocks, trying to find this guy. And if anybody has a question on how many divers does it take to catch a holotype, it's 8. Okay, so this is the last dive of the trip. So on that second dive, we find the initial phase. We get great pictures. We catch a lot of specimens, but it's the initial phase. So if you're going to be describing a new species, you want all phases. You want the juvenile, the initial, and the terminal phase, because wrasses in particular change their color patterns dramatically. So you don't want to just describe it off the initial, because then the terminal could look totally different. So while we're all down there scrambling after the initials, Dr. Sanchez sees the terminal. He goes, I couldn't catch it. It was so fast, but I saw it by this rock. All right, so now we're on the boat. This is the last day. We've been doing deep dives. This is like 60 to 80 feet. Been down there for a while, and we're all looking at our dive profiles. We're like, Dan would tell us not to do this. We're 30 plus hours away from the mainland. And so we're like, all right, we're going for it. So we got a team together, and I have to call out on the far right side, on the left-hand side, that is Bill Lutt from the Natural History Museum, Los Angeles County. And on the right is Ben Frabel from the Scripps Institution of Oceanography from UC San Diego. The other scientists, Ross, Carlos, said, you have to catch it. Here's your spears. And don't come back if you don't get it. All right? All right. So these guys were like, oh my God, I got to get this 6-inch fish that swims like it's on acid and just like all over the place. You've got all the other divers, and the rest of the scientists and myself and Sarah, we went to go dive someplace else to go take pictures of damselfish. All right, because they were only found in that particular spot. So these poor guys had to represent for the scientists, and they've got this huge pole for a small fish. So the team gets in, and that's the only objective. Find the terminal phase male, capture it. Don't even worry about getting pictures, just capture the damn thing. We need this to be able to introduce it to mankind. So they jump in, and I believe Ellie was the first one that saw it from the surface. So she's pointing her finger at it. They go down there. Bill does a great job. Hits it with a spear, but it gets off. Oh my God. Can you imagine? Right? Thing darts down, hides under a rock. So it is like it gets bombed. There's like all 8 people around this rock trying to make sure it doesn't dart out in a direction and get away. And I'm going to share the actual capturing of the holotype with you. So they've got it out from the rock. You've got Bill on the left. He's got it in his hand. Ben is pulling out the Ziploc bags. and look at this handoff. So it's like, okay, is it all the way in there? I zip it, really get it. Make sure you got it. Is it in the bag? Yes, it's in the bag. Grab it, screaming all around. We captured the loose plastic bag, so we didn't contribute to the garbage. But it took 8 people to catch that fish. And everybody was so stoked. And then they were mean. Okay, so Carlos Sanchez, our scientific trip leader from Mexico, he wasn't on this dive. So he was with the other scientists at the other shallow spot. These guys got back first because it was deeper. And they were saying, oh, we didn't get it. It got away. Yes, they did. They did. So they were keeping it way underneath the table. So, we're all coming back. Did you get it? No, and I'm like, ugh. So they were really nasty about it. But Carlos actually wrote this. This is the jar label that goes in the jar with the holotype. So forever and ever, that will be in the jar with that holotype. That's Dr. Robertson on the right. And he was teaching all of the other scientists how he paints the fins of the fish with formalin, which causes them to stay erect. So you can count every single fin ray. So once it dried, we actually had these tiny little mini tanks that we would put the fish in, and we would take photos against a white and a black background, and we'll show you that so that you could see that clearly. But that's as close as I'm going to show you of what that fish looks like. All right, and here's everybody celebrating. So Dr. Sanchez, Dr. Robertson, Dr. Victor. I mean, this was a really big moment. I mean, imagine you are a scientist, you've dedicated your entire career to marine science, and you get to discover a new species. So really, really big event for all of us. Okay, so moving on. So we created a library of over 5,500 images amongst all the different photographers over this 10-day period. We really focused on taking lots and lots of images of the endemics, lots of different individuals, because we wanted to really make sure we got the color pattern by island. Remember that. We're going to come back to that. All right. We were looking for the really cryptic. So this is a reticulated sole here. And this one is so juvenile, you don't even see the color pattern yet. Clingfish we were looking for and collecting. And this is what it looked like trying to find it. So you were sticking your head in all the nooks and crannies going along. And I've got to call out Jeff for that upper right-hand picture. Those are the fantail pipefish. That's a pair. And so they're swimming upside down. All right. And the one on the right is a male, and those are eggs. So Jeff nailed it. These things are really hard to get in focus. And the fact that he got 2 of them and an egg-bearing male is quite the accomplishment. We did not collect that pair. So they are out there repopulating the reef.
Which island?
To be honest, I don't remember which island that was in, but it's in there. I mean, one of the things that we had to do, and I'll show that in a minute. So then we were out in the blue. So this is what most scuba divers go to the Revillagigedo Islands to see and do. So you've got all of these big megafauna opportunities. Spent a lot of time, if you've ever dove with us, in the rocks and sand. And if you look closely, you're going to see some of our mascot fish down there at the bottom. You can see the little white splotches being on their back. So looking for fish that you'll find in that habitat. So tiny little fish. Yeah. Spend time looking, and on the far hand right, you've got Carlos. So why don't you describe what's happening there?
So there's a lot of fish who are very light sensitive. If you put a very bright light on them, they're gonna take off. So it's impossible to focus on a fish if you don't have some sort of light on it. So what we do is we use a red light. Red is invisible to a lot of the fish and to us as well. It's red is the first color that disappears somewhere around 15 feet in the water. And so by using a red light, we can get a little closer to a lot of these fish. Now, it doesn't mean that they don't feel it, because it puts out some heat, but we can get some better photographs because of it. And we spend a lot of time shooting with red light.
But it's a challenge. You're shooting a red fish with a red light in a dark spot. It's really hard to see them and for the camera to focus on them. All right. And then here, Carlos, you can describe what we're doing here.
So this is at, I believe this is in San Benedicto. So we're going after the clingfish, right? So we're being thrown around. Look how far out of the water the clingfish is. So I found this little trick of just putting the net under, instead of trying to swat it like you would a fly, just put it under. they'll kind of jump. They jump for their life. And the first time I tried to catch one, it jumped over my head and out that way. I realized, I got you. So that's how you guys get away. All right, so we're doing this over here. About 200-300 yards away are the scientific team, and they're collecting. They're shooting stuff. They're stuff. And when we all get back to the water, Dr. Geithner, who is amazing with a pole spear, she had very few fish that she brought back. And we go, well, what's going on? She goes, well, I stopped shooting when the 12-foot tiger shark swam over me so close to me that I could have reached up and touched it. All right. So, and we had no idea the fish was there, and we're over here playing with little clingfish. Meanwhile, there's some monster, somewhere close by. Luckily, we were never bothered. But you gotta kind of put that in the back of your head and go, okay, it's not there, it doesn't exist, I don't see it, it's not there, or else you would never do this.
I can't see it, it can't see me.
Exactly. All right, so fun was out in the water. Here comes the work. So at the end of the day, all the photographers would get together and we'd have our cameras and our computers out. We're downloading our images and we're having to name them. And that was quite an experience because you had to put the scientific name, you had to put the common name, you had to put the island, you had to put your initials. So that way we could search all these images after the fact. So here's the group of photographers all working hard. And a lot of these species we weren't that common or familiar with. So having to help each other make the ID. And making the ID was not always easy. I mean, here's one of the things that happened to us. So on the left, this is the flag cabrilla. It's known pretty much throughout the East Pacific, with the exception of Clipperton Island. And on the right-hand side is the Clipperton grouper, same genus, only find it at Clipperton, Revillagigedos, and some spots in Baja. All right, so you look at it and like, okay, they're pretty different. I think I can make this ID no problem. Get out there. Okay, here's the representatives of the family. And on the right-hand side are fish we were finding. So is that a Clipperton? Is that a flag cabrilla? I don't know. Someone tell me. I need a name. So you would get the scientists got into some pretty big debates on which species it was or they thought it was. And ultimately, we're not even sure what's happening here. We don't know if this is hybridization. We don't know if when these species were described, there weren't enough photos, so they didn't take into account the variability of the different color patterns. So we're not really sure what's going on. So there's work that's going to be done on the DNA side along with our photos to try and see if they can tease out what's happening. Okay, so, and here's another great story. I love this one. So this is the spinster wrasse. The region is the entire tropical Eastern Pacific. Now, this fish was collected in the Revillagigedo Islands, specifically at Socorro, back in 1882, and it was described, just like I showed you that black wrasse at the very beginning. And because people that go to the Revillagigedos are going to take pictures of sharks and manta rays, no one's taking pictures of the wrasses. So the team goes out, we're taking pictures of everything. And actually, got ahead of myself. So these are the spinster wrasse throughout the tropical Eastern Pacific. So the first row is from Baja. You've got the juvenile, the initial, and the terminal phase. These are from Panama, and this is the Galapagos. All right, so this range is correct. These guys all look the same, right? Bottom row, this is what we were taking pictures of in the Revillagigedo Islands. That's really different. All right, and so the scientists are like, what's going on with the spinster wrasse? I mean, this is consistently different. We were not finding the patterning that you found in the initial and the terminal phase and the other region in the Revillagigedo Islands. So now what do? Well, DNA analysis has to happen. You got to look again at the morphological differences, but it is highly likely that the wrasse in the Revillagigedo Islands is the spinster wrasse because that's where it was first described. And the one everywhere else is a different species, and we've been getting it wrong for 140 years. All right, I mean, doesn't that just blow your mind? So this was a really cool part of doing this research, getting all of these pictures to be able to help them look and start noticing, wow, this is different. All right, here's another great example. I got a whole bunch of reef fish ID people in here. How do you tell the difference between these two species? Any guesses? It's hard. You guys see anything obvious?
Yes.
Scales. Good job. Very good job. Yellow soldier fish. 3.5 scales above the lateral line. Okay, I hope they stay still for you. All right. Panamic soldierfish, 2.5 scales above the lateral line. So before we went there, Dr. Robertson was going, okay, both of them have been reported in the Revillagigedo Islands, but these are fish that are really easy to confuse. So are they really both there? And this is where photography really comes in, because between the 11 photographers, we were able to take pictures of 69 different individuals in this family that we found. All of them were the yellow soldierfish. So based on our experience, we didn't see this on any of the islands we went to. Dr. Robertson also contacted the specimen museums that had this genus from previous expeditions and said, hey, please go and validate that every single one of your specimens, which species is it? How many scale rows above the lateral line do they have? Is it the yellow or is it the Panamic? So the first round of answers come back, and over 250 individuals that had been collected by previous expeditions all came back yellow soldierfish. So now we're thinking, all right, those guys got it wrong when they said the Panamic was there. But then more results kept trickling in, and a Canadian expedition in 1968 got some small, sort of middle initial phase specimens that were the Panamic. So this becomes really interesting when we're looking at that connectivity. So are these fish actually making it from the mainland over to the Revillagigedo Islands? So that's part of one of the questions that we had on what's going on between these big distances and fish being brought over.
Or were they there and now they were no longer there?
Yeah, that's another thing. That's a very good point. Okay, and then you can see, I was really happy when Bill and Ben sent me pictures of their labs, because that's what the specimen labs look like, and that's one of the microscopes they use to be able to look closely at the different scales and the fins and everything to be able to make different IDs. All right, so the other thing we wanted to do was collect specimens. Here we have part of the scientific team. They had all the pole spears heading out, and they would go out and collect. On the right, we have Dr. Robertson out there collecting, and there is Fernando. The master student gets to carry the dead fish. All right, so his job was to look for the sharks and not give up the collected specimens to the sharks. So Carlos was like, drop it. If the shark comes, drop it. Forget about it. I'll talk to Ross.
I actually pulled him apart, because Ross would never let go of the fish. Knowingly, he would not let go of it. I think the shark would actually have to pull it away from him before he would let it go. So I told him, listen, your life is not worth it. We can always get more. Just let it go if the big animals come around. I'm going to tell you a little story about the dive guides. Okay, so when we were going out to these islands, all the dive guides had dived the main 3 islands, all except Clarion. So Clarion was blank slate. Nobody had been there except for the captain and for Dr. Sanchez, something like a decade before. It had been a long time. Everybody was going, and we were all doing exploratory diving. Knowing this, Dr. Sanchez, Ross, Allison, and I pulled in the dive guides. Normally, if you've ever been on a dive boat, a liveaboard, or even a resort, they always tell you, they give you a dive briefing. They tell you how things are going to work. This is what we do. This is what we expect from you. This is what you should expect from us. But we reversed it, right? So we said, sit down. We're gonna tell you how this is going to work. Okay? You are not dive guides. We are not following you. If you try to get us to follow you, you're going to be herding cats. Okay? The guys who are shooting photographs are going to find something under a rock, and they're going to just sit down and photograph it, and the rest of them are going to go someplace else. They are responsible for getting their butts back to a safety stop and getting up on the boat. That's their job, right? And if they're buddy teams, they should at least take care of one another. Your job is to hover. You're going to be hovering, and you're going to see 2 over here, 1 over there, 2 over here. And if they ask you for directions, point. All right. And if you see somebody in trouble, of course, give them a hand. But you're not guiding. Okay. Let's get that straight, because if you do, you're going to be very frustrated, and people are going to be very mad with you if you tell them, you got to come this way. So that's what we were doing. On top of that, the dive guide rover satellite dude who was with the collection agency, your job also is to keep the sharks away. So all right. So that is what we told them when we first got on the boat, and they also went, Oh, jeez.
But we really have to compliment the crew because they really got into what we were doing. They were actually, the panga drivers learned how to take the specimens from us and put them in the bag. So they were highly involved. We had the people on the boat coming up, watching us while we were processing the specimens. The dive guides started getting into it, going, okay, what are we looking for? And they started to try and help find these fish. So I really have to compliment the dive organization because they really bent over backwards to help make this expedition happen. And the food was amazing. So they kept us very well fueled to get out there to go dive. All right, so you were asking about angst. Okay, the angst was there. So part of science, you need specimens. You need to get enough specimens so that the statistics support it, right? So part of what Michelle was doing on looking at this DNA and trying to see how long, one, are they still sharing DNA? Are they not sharing DNA? And how long has it been? Is she needed to get a lot of individuals of the same different species. And so that's part of what they were doing. So we collected 900 specimens, fish specimens. Those were divided between the 3 organizations. So a third of it went to the Universidad Nacional Autónoma de México, which is their collection for the entire country, their specimen collection. Another third went to the Scripps, and then the other third went to the Natural History Museum. And so all of those have been documented. They are now available. So if you're a scientist, you can do a search. You can find out where they are. You can check out a fish and have it shipped to you. So you can do your research and then you ship it back. So it's super cool. All right. So and Carlos mentioned that is the fish processing table that was set up. So it was the 4 scientists were always late to dinner because they had a lot of work to do once processing the fish. Here they are hard at work. You can see some of the tools of their trade, and they would actually tag each fish. They had a number. The number was written down. It was all very well documented. Here you had a microscope so you could get close-up looks. A lot of fun putting this together. And here you have the whole process. So the fish would come in, it would get tagged. So you would actually sew in a tag onto the fish so that it had a number. They would write the number down. They would take a DNA sample so that they could then have that ready to go. They had, what else were we doing?
So
and then
logging it into a spreadsheet.
Yeah, putting it into the spreadsheet, where we found it, the dive. I mean, all very detailed. And then the photographers would be up there drinking beer watching. All right. And then once they would get the specimens processed, we were put to work again. So we had these little tiny fish tanks, and we would put the specimens in when they were still very fresh. And we would put a black background and a white background and take pictures of them so that you could really see the fine details and the color patterning. And so again, these were also being captured so that we have a lot of data. So we didn't get to drink a lot of beer. All right, so we had 522 tissue samples that were taken to be sent away for DNA analysis. Over 100 species were collected. Again, we're waiting on the DNA to come back to be able to finalize exactly which species were collected. And this is the importance of having specimens. So why do these museum collections exist? Well, the museum collections the specimens are actually documenting what was happening at that point in time in that location. So think of every single one of those fish as a time capsule. And things are changing on the planet a lot. And so you need to constantly be adding more specimens so that you can document those changes over time. And as Bill was explaining to all of us what his job is and why specimen collections are so important, one of the stories he shared with us, which really resonated with me, was the fact that they had a student, a marine biologist, doing a study on microplastics in fish. And the Natural History Museum has thousands of specimens, some of them 100+ years old. And so she was able to go in and pull out these specimens and look at the stomach content and be able to say, hey, before the 1950s, no microplastics into fish. And then after 1950, you start seeing them build and build and build. So this is one of the reasons that these specimen museums are so important and what they add to our knowledge base. This fish, this is one of the fantails. It was not the pregnant male that Jeff took a picture of. It was one that we collected, and already a scientist has requested it as part of a study they're doing on the genus, on the Doryhampus genus. So already the specimens we're collecting are being used by other scientists for their studies. And, all right, so the black and white mystery wrasse or holotype, it is in the museum. It will forever be there as the representative of this species, and it's available for scientists to pull so they can compare it to other specimens and see how is it different or how is it similar. All right. Something else we were doing is we were documenting fluorescence. So we had in one of the rooms, we had to take over someone's sleeping room. We put in the fluorescent light. So we get the specimen that would get cataloged, and then we take it downstairs in the black room, turn on the fluorescent light. And here's a great example. So this is a calico lizardfish half buried in the sand. Ellie's good picture. That's what it looks like captured with a white background on the boat. And then on the far right, that's what it looks like under a fluorescent light. I mean, check out that patterning on the head. Isn't that super cool? I mean, actually super cool.
It's still viable. It's alive.
At that point, I don't think it's alive.
So, right, so it's not
Okay.
So the color might be affected by whether they're alive or not?
Good question. In fact, we don't know all the answers. This is a brand new area of study. They've been only studying it for the last 10 years. And there's a lot of theories as to what's happening. So one of them is they have fluorescence because there's a lot of fluorescence. We don't see it. Our eyes can't see it. But there is a lot down there. If you ever go down and do a night dive with a fluorescent light, you see all the cool colors. So is this a way to camouflage in this very chaotic environment? Because if you didn't fluoresce, you'd just be this black spot, right? And if you're a smart enough predator, you could recognize the shape and get eaten. Another is they use it to attract mates, or at least to differentiate between the sexes. That's one of the other hypotheses. One of the ones that I read what I thought was really cool is there's some that have a lot of red around their eyes, and they think they might be red flashlights that the other fish don't see, and it's a way for them to light up and find prey. I don't know how they're going to prove that, but that's pretty cool. Then that might be of no advantage or disadvantage. We don't know. So there's a lot of research that needs to go in. And what we were doing is at least trying to create a baseline of which fish fluoresce, how do they fluoresce, and what colors they fluoresce.
Oh, I guess you answered it. I was thinking like the flashlight fish that use like to attract.
To attract.
To communicate or something.
Exactly. So this is a brand new area of study. So people here in the Caribbean, if you want to take fluorescent pictures at night, there's not a lot of pictures in the scientific database, so ways we can all contribute. All right, the other thing that we were trying to do is to understand the diversity, abundance, and dispersion of fish eggs and larvae. So we were doing zooplankton collections. So we have Dr. Sanchez and Dr. Victor. So Ben, every night, he'd go to the back of the boat, get his bright light, sit, and there's sharks, a lot of sharks, all right? So he's got his arms over the side, and when you can see sharks underneath, you can see the shark in the back. You did not want to stick your hand or your foot overboard because they were there just waiting for one of us to fall in, and then we'd get all of us back there, and I mean, it was like everyone was holding on to everybody else's belt buckle as the others were trying to collect some of these things that were going by, and we caught really cool things. So this is the first time that the white-edged moray larva has ever been documented in the Revillagigedos. It's completely translucent. And on the left bottom side, it's where its eyeballs are, and it's sort of pulling itself backwards. This is the largest eel larvae in the New World at 5 inches. And then in the center, you have an example of other specimens collected elsewhere, what they look like. So their body becomes white once they've been preserved. And the one on the far right is the one that we collected. Yeah.
So bad.
So really cool stuff. And here we have thousands of larvae that were collected. So they pulled the zooplankton net. And I mean, I've never really seen this, so it was all new for me. And what was happening is in the collections, you'd get a lot of crustaceans. So they would actually have to pick out the individual fish larvae from the crustaceans. And so you can see that on the far hand right, the fish larvae and the crustaceans around them. And I am glad I'm not Ben Victor, because this is the work he has to go through in processing all of these specimens, doing the counts, how many of what. And I know, Carlos, you were going to mention.
So one of the things that surprised not just us, but surprised Ben, is that on different nights, successive nights, one night you were collecting tons of small pufferfish, the larvae of this pufferfish. The next night, no pufferfish, nothing but lizardfish. And the next night it was something else. And it was very clear cut. It wasn't like they were all mixed together. This was, one night was this, one night was this, one night was this, which was completely surprising and unexpected. And so, now why is that happening? How is that happening? We don't know. And how do they coordinate who gets to go next, right? So we thought it was very interesting. Lots of questions, more questions I think that we had and that need answers from this trip.
All right, and so the final objective was to determine the diversity and connectivity between the Revillagigedo fish island population and the other East Pacific populations. And this is where Dr. Gaither from the University of Central Florida was her main focus. And she really needed to collect, she's been collecting DNA from all around the Pacific, both the East Pacific and the Western Pacific, so that she can have the DNA to be able to do these comparisons. And the questions that they're trying to answer are individuals crossing that Eastern Pacific barrier? Are they able to make it across those thousands of miles in 4-mile deep water? If so, which species are making it across on a regular basis? Which direction are they coming? Are they going east to west or west to east? If it's not occurring, how long ago did it stop? All right, so very interesting questions. And here's an example of the currents. So from the Revillagigedos, the currents are flowing to the west. So it's much more likely that species from the Revillagigedos are being carried west to the Hawaiian Islands, which would be the first place that they would land. Whereas further south, the currents are heading to the east. And Carlos and I were in the Galapagos in 2021, and that is actually the elongate surgeonfish, and that was the first time that species has ever been documented in the East Pacific. So it is brought over from all over way over here on the other side of the East Pacific barrier to the Galapagos. And in the photos, you could see several individuals of that species, but we don't know, were these just migrants that were brought over? And is it a big enough population that they can establish themselves and now the species exist on the East Pacific, or are they going to die out and that'll be the end of them until another group potentially gets brought across? So very interesting. All right, so I mentioned this earlier, remember this point. So we were taking pictures of the endemics in particular on every single one of the islands, trying to get as many individuals as possible. And as the scientists are looking at all of these hundreds of images, they're like, hey, wait a minute. The Revillagigedo barnacle blenny photos from Clarion, the most western Pacific island, look a little bit different than the photos that you guys are getting of the same species from the Socorro and San Benedicto Island. Are we seeing microendemism happening? So is that 250-mile divide enough to allow speciation to start occurring within the Revillagigedo Islands? Again, waiting on DNA to see what it says. They're going to have to look at all of that, look at the morphological differences, see is the patterning consistent enough and the DNA there to be able to say that, yes, this is happening, or no, it's not happening. So a lot of really cool science being done. All right, so you got to be tired. I mean, 10 days of this is a lot, right? So where we are now. So a lot of work is in process. We've got DNA processing going on over 500 specimens. We're processing 1,000+ larval specimens that have to be done. The museums are cataloging all the fish that they received, putting them into the database, making that database searchable on the internet. The museums are still going through all the previous collections and getting back, validating that, yes, that species is that species, so it is a true voucher. A lot of work being done by the scientists to look at all of this information, figure out what it says, and start writing the papers and to describe the species. And finally, we're waiting on it. They'll start publishing it. And once it's published, we can share the live photos of the fish with you. All right. So in summary, this expedition was a huge success, both from a scientific standpoint and also from an experience standpoint for all of us photographers. We discovered at least 2 new species, and we think it potentially could be 3. We've discovered a new genus that was not known to be there, 4 other species that were not known to be in the island chain. We updated the fish census by island, so 152 different species documented within the chain. 1,900 specimens collected between the larvae and the actual large individuals. And we have this image library of 5,500 in situ diagnostic images. So now we can do the comparisons and see, are there differences by island? How do these compare to the other Eastern Pacific? And the great news is there's still so much more to discover. All right. So here we have our photographers celebrating, because for all of those of us who were able to participate, this really was a once-in-a-lifetime experience, and we were all just thrilled to be part of it. So thank you very much.
Sharks didn't collect you.
I know, we didn't bring back any missing limbs or anything.
Well, you didn't tell us that wrestling that tiger shark onto the boat was a big problem.
Yeah. So Jeff and Ann were on the trip with us. They've contributed their images and helping us find it. Is there any other questions that are out there from billions? Yeah, well, okay.
How about one?
What's the water temperature?
Okay, what was the one?
Oh,
So I'm supposed to repeat the question. So what is the
well,
water temperature?
Okay, water temperature. Clarion was 6 degrees Fahrenheit colder when we got there. So it was a shocker. We went from about 79 to mid-low 70s in Clarion. So it was a lot chillier. I checked, actually. I knew somebody was going to ask.
All right. Another question. Yes.
So when people check out the samples, you're talking about them finding the stomach contents and stuff. So it actually destroy that sample when they check them out.
Yeah, so we're getting way beyond my knowledge base. So they are allowed to check them out. I know in that particular one, they were looking at the stomach samples, and that's all I can tell you. I don't want to make stuff up, but that was part of it. So that particular research, they were able to access it. And that some species or specimens, they have a lot of different individuals. So it'd be easier to say, yeah, we can sacrifice that one. And like here, like Michelle, she needed to collect 25 individuals of each of the different species she was studying to be able to have enough for the genetic analysis to be statistically significant. Yes, Mark.
Do you think that macro weather patterns of La Nina and El Nino might affect some of this research, or that you might even see species change?
Can I answer that?
Yeah, answer. You got to
All
repeat
right.
the question.
So the question is, do you think that La Nina or Nino weather patterns can change what you're finding? Absolutely. Absolutely. We saw it when the first paper we published here in Alligator Reef, we saw that there had been a change from 50 years when we did it. So fish that you find one year may be gone the next. Fish that weren't there the year before now all of a sudden are there.
And to jump in, Carlos, one of the species we were looking for was a chromis. And there was a lot of question on, is that fish there? And has it potentially gone extinct? Because it's gone extinct in some of the other East Pacific islands. And one of our friends, Carol Cox, took pictures of it several years ago, and it was on Roca Partida. So when we were at Roca Partida, Ross was telling everybody, get up close to the rocks and look for this chromis there. None of us found it. So the question is it extinct? Is it just the temperatures? Does it want colder or warmer? And interesting, I was on the phone with some scientists from the Galapagos this past week, and the El Nino is happening, and they were saying how it's really warm and there's no fish. So the warmer water was very clear, but they weren't seeing the same numbers of fish that they normally do. So it definitely will impact what's there and what's not there.
So they wouldn't migrate necessarily like birds migrate south and north.
Yeah, I mean,
Temperatures and stuff.
Gorgeous up and down the
Maybe the bigger stuff. But
yeah, or the depth,
yeah.
but food. So, I mean, there's a lot of things that are playing into it, but they were commenting on how the water's much warmer than it normally is this time of year.
So
and the
that's one
quantity of
things
sharks
you
wasn't there.
Have to record.
Yeah.
Because that's changes.
Yeah.
So one of the things Allison said earlier is that this is a very short snapshot of what was there on that date in that year. During that time, you record water salinity, you record water temperature, you record all of these things. And so that's what was there with these conditions. Next year may be different. We don't know. I mean, and one of the things that happens in the Revillagigedos versus say like Galapagos is Galapagos, you have a city or you have population center, and you can stay there and study fish in the Galapagos for months or years at a time. You don't have to leave. You got the Charles Darwin Center there. But these islands are only accessible during non-hurricane season, and they're very far out. So here you have, we actually were on the water 13 days, 10 in the water. There were 3 days of traveling plus overnights and everything like that. Our time in the water was really limited. So the amount of information we can gather on a trip like this, while it looks like a lot, it's probably not that much. So we would really need to visit these islands year after year, or month after month, to be able to get a better, bigger picture of what's happening. Does that answer?
Yes, in the back.
Any night diving?
No, they didn't let us night dive. I mean, you saw the sharks too. It was like, all right, I can't see it during the day, but at night they would circle the boat and they were chasing the fish around. I mean, well, yeah, and that's one of the cool things with the zooplankton, because they're catching all the larvae. And so there's species that we collected with the zooplankton that we did not capture during our daytime collections. And when we were first putting the trip together, I was talking to Jeff about, hey, let's, can we do the black water diving and all of that, but they don't allow you to do night diving out there. So, and to be honest, we were wiped out. I mean, by the time dinner was over, people were pooped.
Oh yeah, for the population connectivity, do you happen to know what approach for genetic distance the researchers are using?
I'm sorry, you are way over my head. I can advise you. Go. Dr. Gaither has her own lab and it has a website, and I actually read it, and I read it again, and I read it again, and I finally called her and I said, okay, what do I put on your slide? So she has a lot of really cool information there, and you will understand it a lot more than I will. So
but I will tell you something that I've had the conversation of what is a species a lot of times with different scientists, and all you have to do is you put two scientists in a room, ask them what a species is, and you're going to get a food fight. Absolutely. What constitutes a species? How different does it have to be? Is it 1%, 2%, 5%? And then what's the morphological difference? Can it be a color variation? Does it have to have an extra fin ray or something like that? So that is a big question, not easily answered by people who we play at being scientists.
Other question.
Are those 5,000 or so photographs that were going to be available to the general public?
Well, so we all publish them on our own. I know Jeff has a website. We have a website. Dr. Robertson at the Smithsonian has all of them. And so he is actually loading a lot of them into the Tropical Eastern Pacific AI to help train the AI on how to recognize fish, so we can start loading pictures, and the AI will tell you what you got a photo of, as well as in his Fish ID website and app. So the app is going to be updated soon for both the tropical Western Atlantic and the tropical Eastern Pacific, and a lot of these images are going to be included.
What I did do, should have done is we should have put our website address where you can go to our galleries and you can go to the Revillagigedos and you can see all the photos that she and I took.
Yeah. Okay, we'll give that out in a minute. Good question, though. Yes, Nate.
Did you guys get confused with somebody else having the same?
Oh yeah, it was well, like the poor dive guides. I mean, I got to give these dives credit. We get on board the first day and we're like, okay, great, you're not leading. You're just there to get us all back. And in the first dive we went on, our dive guide on our boat, he's like, yeah, everybody gets in the water and then they're like gone, every direction. And our boat was particularly bad at it because Ross was with us, so he's out spearing. We were with Jeff and Ann and Carlos and I, and we would get caught up in what we were doing, and we were gone. You just see our fins taking off, so that he would be sitting there, just like, yeah, they're everywhere. But the great thing is everybody was super experienced, so everybody was able to pop their own SMBs, get their way to the surface safely. And the other photographer team, I have to give them points because they all at least swam in the same general direction. I think when we were on Rocca Partida, Jeff went one way, we went the other way, met on the other side of the rock and kept going around. So yeah.
Did you guys sleep?
Yeah, we did. I mean, but we were so hyped up. I mean, to be exposed to this, to see true marine science being done. I mean, it hurt to see the fish being collected. It truly hurt as fish aficionados. But then understanding the science, understanding the importance of contributing to the museum collections and how those fish will be there, not only to document what it was then, but to help us understand what we need to do going forward. So yeah, it was a thrilling experience. All right. Anything else? You guys have hung in there. Yes, Lillian.
It sounds like you collected a lot of information, a lot of specimens. Who's going through all of that now? Is it students at different places?
It all depends on where it was sent. So in the case of the specimens that are staying in Mexico, I know that in talking to Bill, Ben, and Dr. Carlos Sanchez, Dr. Carlos Sanchez is a professor, and he has a very large group of not only doctorate but master students, and they were getting to do a lot of work from what I understand. So processing on making sure that everything was properly named, helping separate them out. Exporting fish is a big deal. You've got to get permits. It has to be exported in a certain way. So they were very instrumental in helping make that happen. And then once all the specimens got to Scripps and got to the Natural History Museum, each location has their own way of handling things. So that also had to be done locally, put into their system. And the cool thing with the Scripps and the Natural History Museum is they then upload their information to a global internet database that makes it searchable. So scientists can find out what they have and then they can request it. Yes, ma'am.
Just a quick comment and a plug for the LA County Natural History Museum. I haven't been there in years, but for those of you who participated in the City Nature Challenge in 2023, which collected over 57,000 photos of different species all around the globe, that was started by the LA County Natural History Museum in competition with San Francisco. And now involves people all over the world taking pictures for 2 days. And they all get uploaded, and that's all out there on the web. You can see the list of all the species. Here in the Keys, we did really well. We were in the top 20 for almost all 3 categories: number of participants, number of species, and number of observations. So,
Lori, send us the link.
Yeah.
Yeah,
Or
just
post
put
it in
in City Nature Challenge 2023.
And I think what Lori's mentioning, the amount of contribution that those of us who are citizen scientists, reef members, photographers, people that will go out in their backyard and take pictures. There's so many ways that we can contribute. And it is so exciting, to see, for Carlos and I, we wanted to be marine biologists as kids, and to be able to go out there and to contribute and to know that our images are really making a difference. It's phenomenal. And we can't answer the DNA questions, all right? We can't answer the El Nino and La Nina questions to the depth that we would love to, but we can make a contribution, and all of us in this room can do the same.
One of the things that we really enjoy is whenever we photograph something and a light bulb goes on in some scientist, taxonomist head. Oh, I didn't know it could look like that. Oh, I didn't know it was there. Oh, I didn't realize it went that deep or that shallow or this or that. And so our photography adds to this database, and I think it helps fill in all of these missing pieces of the big overreaching puzzle that is life. Yeah.
An intimidatingly intelligent group.
It was, and they were so fun. We had so much fun. It was a great group.
When at the beginning of the trip, everybody would get a chance to go up. The scientists would go up and describe what it was that they were going to try to do during the trip. What was their goal and what were they going to try to find? And we knew they were speaking English. We knew there were words in English. It just spread over my head, especially when Michelle started to talk about what she was trying to study. I was like, wow, okay, all right, I'm gonna take photos. You do one.
All right, any other questions? I know you guys hung in there for a really long time, so thank you for hanging in. All right, and I know